Well, thanks, Gary, and I'm pleased to be here again. And I appreciate the fact that all of you folks have come out to spend the day with us. I think we're delighted that you've chosen to do that. Walt, thanks for bringing a different perspective to our meeting. I appreciate that presentation very much. Should say that I've had the opportunity to work fairly closely with Walt and his colleague Lowell Randall as I've been involved in the policy committees of ASAS and FAST basically since this program started about 4 years ago. And so as I've spent time working with them, I have gained increasing appreciation for what they do for us in Washington, D.C. So we appreciate that. So my task here today is to talk about diet and immunity. Now, some of you know that during the last few years, my lab has had a focus on evaluating things we can do in the diet that can contribute to the health of the pigs. So it's a question of how can the feed be part of the herd health program. Now, if you look, and mostly we're looking at feed ingredients and feed additives, although certainly there are other aspects of the diet that might be considered as well. But as you look at these feed ingredients and feed additives, most of them seem to bring benefits in one of two ways. Either they modify the microbial populations in the digestive tract, or they have an influence on the immune system. Now, as we have worked in this area over the last few years, I have increasingly become more optimistic about our opportunity to bring real benefits to pig health and production by focusing on the immune system more than on the microbiota. I think there are important things to be done in both areas, but I'm at this point more optimistic about what we can do in the immune system. I want to recognize my colleague and the first author of this paper, Dr. Yen-Hong Liu, who is currently a postdoc in my lab and has led some of the projects that we'll be talking about here today. When we think about immune function, and I'll confess at the outset that I don't consider myself an immunologist, but when we think about the immune system, the first thing to recognize is that there are really two immune systems. There is the innate immune system that responds to a new challenge, a challenge that the animal has not seen before. And then over time, there is the mobilization of the acquired immune system, and that's the one that produces antibodies. Now, there are reasons why we would like to shift from the innate immune activity to the acquired immune activity as soon as possible, and we'll talk more about that. So we have the innate immune system, the acquired immune system. The acquired immune system is the one that produces antibodies. The innate immune system produces a collection of responses that collectively we refer to as inflammation. Now, inflammation, as you would recognize, involves several things. First, it involves redness and swelling at the point of attack, and then it involves some behavioral symptoms as well. There's lethargy. There is reduced feed intake. If it's a systemic inflammation, then you probably also see fever, an elevated body temperature. Now, the lethargy, not wanting to move, and the reduced feed intake are part of what is generally referred to as sickness behavior. And I think everybody in the room is familiar with sickness behavior. When you get sick, you want to just lie on the couch and you don't have much of an appetite. And exactly the same thing happens to pigs when they get sick. Now, that's a little bit of a problem for us when we want that pig to aggressively consume feed and grow. And so that's something that we're concerned about. Now, in addition to these clinical signs of inflammation, there are some other more physiological activities going on. One is the production of cytokines. Cytokines simply means cyto means cell and kine means movement, so it's a protein actually that attracts immune cells to an appropriate place in the body where there's an infection. Now there are pro-inflammatory cytokines and there are anti-inflammatory cytokines. And there are some that can't quite decide which camp they're going to be in. Sometimes they do one, sometimes they do the other. But we're primarily interested in the pro-inflammatory cytokines, and that's what I'll be talking about mostly today. There's also the production, and by the way, the cytokines can be made in many tissues in the body. There's also the production of acute phase proteins that are part of the immune response made in the liver. And then there is also, after a challenge, there is the proliferation of immune cells and then the movement of those immune cells to the point of attack under the influence of the cytokines. Now, as I indicated, we have a concern about this package of responses. And that is that first, it reduces feed intake. So that means you're reducing the production of the growth or the production of the animal. Second, we're producing a lot of proteins, cytokines, acute phase proteins, and the immune cells. And those things are all taking nutrients, energy and amino acids that we'd really like to spend on growing. And so we consider inflammation to be an essential part of the protection of the animal, but also very detrimental to production. And so what we'd like to have is a vigorous response at the point of attack so that there's good protection. But then we'd like to avoid the prolonged inflammation that we often see in animals. And so in terms of what we'd like to see in the immune function, we'd like to see, obviously, protection from disease. That's why we have an immune system, is to protect us from disease. But then we also want to avoid excess activation. And in fact, in my view, that's where we can make some real progress in the things we can do in the diet, is by avoiding the excess and prolonged activation of the immune system, the excess and prolonged inflammation, and therefore stimulating more growth. So if there's one thing you take away from this today, I'd like it to be this: the fact that the immune system, while it's very valuable in protecting against disease, can also be very detrimental to animal performance. And one of our goals is to reduce that prolonged inflammation. And so I'd like to show you some data we have on 3 different feed products that, in our hands, seem to have some effects on the immune system, some effects that may be important. Now, we don't have enough time to go into all of these in detail, so what I'll be showing you is just a 30,000-foot view of what these products can do. So we'll have a very quick look at these responses. So first we'll talk about yeast-derived mannans, then a bit about plant extracts, and then about spray-dried plasma. We don't suggest that these are the only products that have effects on the immune system. These are some with which we have experience in our lab. The products, the yeast-derived mannan products we've worked with are the Alltech products, Biomoss and Actogen. Biomoss you may recognize as the original mannan oligosaccharide product. It was originally developed with the idea that it could influence microbial populations in the digestive tract, and it does that. We have become increasingly of the opinion that the more important effects of this product are on the immune system rather than on microbial populations. Actogen is a newer product. It's a more refined product. It contains, just as an overview, it contains a higher concentration of mannans, of mannose, and a lower concentration of beta-glucans. but both contain both of those components. Now, one of the things we've done with this experiment, these products, is some studies with cell culture. Now, I would have told you a few years ago that there's no way I would ever do cell culture, but here I am doing cell culture because I think that's the way we need to get some of the answers that we need. Our approach here has been to take some pigs, kill the pigs, take out the lungs, pour in some buffer, squish it around a little bit, pour it back out. And when we pour it back out, we've collected a lot of immune cells from the lungs, mostly, several types of immune cells. But then we grow them in culture under various conditions and see what happens. And here's what happened in this case. So we're looking here at the production of a pro-inflammatory cytokine called TNF-alpha. And we have first our control cells. Then we just put the moss on the cells, put it in the plate where the cells are growing. And that stimulates production of TNF-alpha. So it activates the immune cells. Then we use this thing called LPS, lipopolysaccharide. Lipopolysaccharide is part of the cell wall of some bacteria such as E. coli and Salmonella. But one thing we know about it is that it fires up the immune system. It dramatically stimulates the immune system. As you can see here, when we put it on these cells growing in culture, they produce much more of the pro-inflammatory cytokine. And then, this is interesting, when we add the moss on top of the LPS, it brings the inflammatory cytokine back down. And so we get different responses to the moss depending on whether the cell is in a control status. In that case, we get an increase. We stimulate the immune system, but when the immune system is already stimulated, we dampen it. Now, you can do the same thing by feeding. So we fed moss to pigs. We took their immune cells. We added the LPS, and the cells from the pigs that were fed moss produced less of the TNF-alpha. And so that's very consistent with what we see here. Now the next step was to take these products into some disease challenge studies. And we're fortunate to have at the University of Illinois some very good, very sophisticated disease containment chambers. We have 16 total chambers. We have 4 pens for individual pigs in each of them. So we can do studies with 64 individual pigs. We actually, what you're looking at here on the left is a hallway, and there are 8 chambers off this hallway. There's another hallway that's identical. But that's where we do our disease challenge studies. We have models for an E. coli challenge and for a PRRS challenge, and I'll show you data from both of those challenges before we finish here today. Here's a PRRS challenge in which we fed the moss product. And here we're looking at serum levels of TNF-alpha, of that same pro-inflammatory cytokine. Now, with the solid lines here are the non-challenged control pigs. And you can see, as you would expect, when there's no challenge to the animal, these pro-inflammatory cytokines are at pretty low levels and constant levels. But when we challenged them with the PRRS, we got an increase in the amount of the pro-inflammatory cytokine. And then here in the second week after the challenge, there was a big difference between the treatments. So the pigs fed moss had much lower levels of TNF-alpha when they were challenged than did the controls, completely consistent with what we saw in the cells in culture. In these same pigs, we measured body temperature, which may be considered a measure of inflammation. Here the solid lines again are fairly flat. I'm not sure what happened at this point. That's been confusing me forever. But no real effects on on temperature in the non-challenged controls. When we challenged them, temperature went up. Not a lot, 0.5 degrees Celsius, at 3 days after challenge. And then in the control pigs, that temperature stayed high for a couple of weeks. But look what happened when we fed the moss. By day 7, it was back down to normal. So suggesting again that there may be some dampening of the inflammation by feeding moss. And we don't know whether that means it's helping the animal recover faster. Don't know at all whether that's what it means. Here's something that I think is very interesting. Here we're looking at white blood cell counts, leukocyte counts. In the same experiment, again, the solid bars here are the non-challenged controls. The hatched bars are for the pigs challenged with PRRS. In all cases, the yellow is for the controlled diet and the blue is for the moss. Now, one of the things that you always see, and we've seen it in all of our experiments, that you see with PRRS is the opposite of what you expect. Because normally when you have a sick animal, the first thing that happens is white blood cell counts go up. But with a PRRS infection, it goes down. And that happens routinely. And so you can see here that at day 3 post-infection, white blood cells are lower with the infection than without. That's the same thing happens at day 7. By day 14, you get the response you would normally expect, which is a higher level of white blood cells. And so we suspect that this reduction in white blood cell counts with PRRS may be one of the reasons that PRRS makes pigs more susceptible to other diseases, to secondary infections. Now, the thing that we find interesting here is that feeding moss increased white blood cell counts. Now, it didn't increase it just in the challenged pigs, it increased it in the non-challenged pigs also. But what that meant is that we never get as low after the infection. And maybe, this is speculation, that helps to protect the pig against those secondary infections. Here's another thing I thought I would never do. Gene expression measurements using microarrays, where we can measure the expression of several thousand genes all at the same time. This is what the people in the field call a heat map. And basically, this bar at the bottom tells you what it means. It means red means that the gene is upregulated. Blue means that the gene is downregulated. We have here a listing of several of the genes involved in the immune system, in the immune response. Here's the control. We feed moss, we get an upregulation, just like we saw in the cell culture. But when we add the moss to the PRRS challenged pigs, we get blue, which means we're downregulating those genes. So these data, it seems to me, are completely consistent with what we saw in the cell culture and in the disease challenged pigs in the clinical responses, saying that in the normal pig, the product activates the immune system. In the challenged pig, it dampens the immune system. Now, one of the questions, so this is a summary. In the healthy pig, it stimulates the immune system. In the diseased animal, it reduces the overstimulation. Now, there are other products that seem to show the same general pattern, but it's clearest. Of all the data, all the things we've worked with, that pattern is clearest in the case of the biomass. One of the questions is, what does that do to disease resistance? Because we're concerned that if we dampen the immune system, that might make pigs more susceptible to disease. What we're looking at here is antibody production at 7, 21, and 35 days after challenge. We're only showing the challenged pigs here because, of course, the non-challenged pigs didn't have antibodies. At day 7, as you would expect, they're not positive. Generally, you expect a value of 0.4 to be necessary for a positive response. So it takes longer than that to make the antibodies. But at day 21 and at day 35, we actually have more antibodies, more PERS antibodies in the pigs that were fed in this case, the Actogen, than in the control animals. So we suspect that that means that we're not interfering seriously with the disease prevention. Okay, let's move on to a second category, and that is plant extracts. And I should tell you that all the work with the mannans was done by Dr. Tung Che, who was a PhD student and then a postdoc in my lab, and he's not here today because he's now an assistant professor at Nong Lam University back in Vietnam. The next set of data I will show were produced by Dr. Liu, who's the first author of the paper. Tip, this is in here for you so we can connect it to food. We worked with 7 different plant extracts, and I'll see if I can get these right. There was carvacrol, which comes from oregano, cinnamaldehyde from cinnamon, eugenol from cloves, anethol from anise, turmeric oleoresin from ginger, capsicum oleoresin from peppers, and garlicon, of course, from garlic. So we worked with these 7. The ones that are called oleoresins are actually extracts, standardized extracts, whereas the others are all synthesized compounds. The first thing we did was the same sort of cell culture study that we did described with the moss. And here we're looking again at the same TNF-alpha, the pro-inflammatory cytokine, only showing the ones that are LPS challenged. There's not room on the slide for the others. But you'll notice that as you go from left to right within each of these sets of bars, we're going to increasing concentrations of the product. And in all cases, as we add the product to the cells, we reduce the amount of the pro-inflammatory cytokine produced, just as happened with the moss. Now, a couple of things here. One is that this says that there's a direct effect on the cell, on the immune cell. So it doesn't have to be a systemic effect. There may be systemic effects also, but there are direct effects on the immune cells. The other thing is, and this is a confession, I'll confess that I'm giving you the simplified version. You know, there are a lot of different pro-inflammatory cytokines, and they don't all respond the same. And so what I'm showing you here is the one that gives a very consistent response. Now, if we've looked at another cytokine or two, and they are influenced in different directions by these plant extracts. We went on to do an E. coli challenge with 3 of these plant extracts. And I don't want to spend much time on this, but just to show you that in measuring the serum levels of TNF-alpha in only the challenged pigs, you see that for the most part, not in every case, but for the most part, those serum cytokine levels were reduced in the animals that were fed the plant extracts. We did another experiment, a PERS challenge, with those same 3, and again you see the reduction in TNF-alpha in most cases when we fed the plant extracts. So we think we're seeing again a reduction in the inflammation. Back to gene expression, but this time I'll present it a little bit differently. What we're showing here is a comparison between among the pigs fed the controlled diet, the challenged versus the non-challenged. And this is in the E. coli challenge experiment. And you can see we have a whole bunch of genes upregulated, a smaller number downregulated. Then only looking within the challenged animals, comparing the capsicum to the control, the garlic to the control, the turmeric to the control. In all cases, we have more downregulated genes than upregulated genes. Now, here's what's important. In many cases, the effect of the plant extracts was to basically neutralize the effect of the E. coli challenge on the gene expression. So in other words, E. coli would cause an increase in the expression of the gene, feeding the plant extract would decrease the expression of that same gene. So it's a partial counteraction of the effects of E. coli on expression of genes of the immune system. We're actually having a lot of fun now trying to follow physiologically just what's happening here with the cascades of immune function. Now, as in the case of biomass, we wanted to be sure that we're not impairing the animal's ability to fight off the disease. And so we have looked at the frequency of diarrhea in the E. coli challenged animals, and we find that there's actually less diarrhea in the pigs fed the plant extracts and having a moderated immune system. In the PRRS challenge, we're looking here at the serum viral load, the amount of viruses circulating in the serum, and this is at day 7 and 14 post-infection. Now, these values are presented as CT values. This is because of the method we used for making the measurements, quantitative PCR. So you get the answer in CT values. CT values are inverse to the actual values. So a high CT value means a low value for the amount of virus in the serum. And you can see that generally, not significant in all cases, but generally we have higher CT values, which means less circulating virus in the animals that were fed the extract. Okay, just in the last few minutes here, and we won't spend a lot of time on this, but we've done some strange work with spray-dried plasma. Here's the logic we started with. There are data that shows that spray-dried plasma reduces inflammation. There are other data that show that inflammation, at least in some situations, impairs reproduction. So we simply put that together and we said, well, maybe if you feed plasma, you can reduce the inflammation and therefore eliminate that block on reproduction, therefore improve reproductive performance. We wanted first to do a proof of the principle experiment. We wanted to just see if that would work without getting into a study that could be practically applied on a pig farm. And so we did it with mice. And so I don't usually talk about mouse data at this meeting, but let me just show you one slide of what we've found. We were dealing with a situation where there was very poor reproduction in these mice. Now, I won't go through the long, complicated, and somewhat embarrassing story of exactly how we created that situation, but what you see here is the pregnancy rate of mice. These were all mated, and this is the pregnancy rate at day 16, sometime in late pregnancy, of these mice that were all fed a controlled diet. And you can see that in this case, less than 30% of them were pregnant, and is more down around 10 or 15% in most of the others. There were 4 separate groups scattered over time, and then the last bar is for the overall. Here's what happened when those mice were fed plasma. 8% plasma in the diet. Dramatic increase in the pregnancy success. Now, that does not in itself confirm the hypothesis that we set out, that plasma reduces inflammation and thereby improves reproduction. But we've gone on and done some other studies, and I won't show you the data, but let me just tell you what we think it means. What we think it means is that what we're seeing in that reproductive failure is a failure of implantation, because we're seeing it very early by day 4 in pregnancy. We also see that the failure of pregnancy is associated with inflammation, In other words, there are higher levels of the pro-inflammatory cytokine. Again, we were using TNF-alpha. Higher levels of the pro-inflammatory cytokine in the mice that were not pregnant than in the mice that were. And we also did see that spray-dried plasma, feeding spray-dried plasma reduced the inflammation. And so our conclusion is that our hypothesis holds, that feeding plasma reduces inflammation, and that allows normal reproduction. One other comment, the data I showed you on the previous slide were from feeding 8% plasma in the diet of these mice. We have since confirmed that we get almost that big as an effect by feeding 1% plasma. And so it's not terribly sensitive to the level of plasma in the diet. Now, what's this mean for pigs? I don't actually know. I don't think we're going to triple pregnancy rate in pigs in most commercial pig production systems. But I do think that to the extent that we're losing pregnancies in pigs for some reason because of inflammation, that maybe the plasma can help alleviate that. Now, I would bring you back to some of the data you've seen that have been published by Joe Crenshaw and others, in which they fed plasma to sows and found, in many cases, a small improvement in farrowing rate. Now, I don't think they've ever seen an increase in litter size. They saw an increase in farrowing rate. And it seems to me that what we've seen here may be an explanation for that. And so we need a lot more information before we can draw that conclusion very firmly. Well, in summary, the immune system is very important to protect from disease. You all know that, but the message I would like you to take away today is that it can also be very detrimental. So excess inflammation and prolonged inflammation is detrimental to animal performance. We're looking for ways that we can reduce that excess prolonged inflammation. And we believe that we have data showing that yeast-derived mannans, that plant extracts, and that spray-dried plasma, at least these three can do that in pigs. Well, thank you very much for your attention.